A nucleotide consists of a base, a pentose sugar, and a phosphate group. A nucleotide consists of a base, a pentose sugar, and a phosphate group. The paired bases project towards the center of the double helix. The structure is stabilized by hydrogen bonds. The structure is stabilized by hydrogen bonds, which are dotted lines between each base pair. There are 2 hydrogen bonds between adenine and thymine, and 3 between cytosine and guanine. There are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine. Nucleotides are the monomers of nucleic acids. This is the phosphate group. This is the dextrite sugar, and this is the hydrogen bond. The hydrogen bond between adenine and thymine, there's 2 hydrogen bonds between them. Between cytosine and guanine, there are 3. Three bonds between cytosine and guanine, Guanin, cytosine, Cytosine, and 2 bonds between adenine, Adenine, and thymine, 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 adenine and thymine. The average gene consists of 3,000 nucleotides. DNA is packaged in the nucleus like this. Packaging of DNA into a chromosome in a dividing cell. Packaging of DNA into a chromosome in a dividing cell. When packaging is complete, 2 identical DNA molecules and their histones form a pair of chromatids, which are held together by a centromere. When packaging is complete, 2 identical DNA molecules and their hormones form a pair of chromatids, which are held together by a centromere. two identical DNA molecules form a pair of chromatids, which are held together by a centromere. A chromosome is a highly coiled and folded DNA molecule that is combined with protein molecules. A chromosome is a highly coiled and folded DNA molecule that is combined with protein molecules. That's the chromatin. That's the chromatin fibre. Gene expression is the process where a gene's DNA is used to direct synthesis of a specific protein. Gene expression is the process whereby a gene's DNA is used to direct synthesis of a specific protein. First, the information encoded in a specific region of DNA is transcribed, which means copied, to produce a specific molecule of RNA, which is ribonucleic acid. Gene expression is a process whereby a gene's DNA is used to direct synthesis of a specific protein. First, the information encoded in a specific region of DNA is transcribed to produce a specific molecule of RNA. Then the RNA attaches to a ribosome, where information contained in RNA is translated into a corresponding sequence of amino acids to form a protein molecule. Gene expression is a process where a gene's DNA is used to direct synthesis of a specific protein. First, the information encoded in a specific region of DNA is transcribed to produce a specific molecule of RNA. Then the RNA attaches to a ribosome, where the information contained in RNA is translated into a corresponding sequence of amino acids to form a new protein molecule. Overview of gene expression. Synthesis of a specific protein requires transcription of a gene's DNA into RNA and translation of RNA into a corresponding sequence of amino acids. Step 1. Transcription. DNA and RNA store genetic information. A sequence of 3 nucleotides in DNA is called a base triplet. The DNA and RNA stores genetic information as a set of 3 nucleotides. Each DNA base triplet is transcribed as a complementary sequence of 3 RNA nucleotides called a codon. Each DNA base triplet is transcribed as a complementary sequence of 3 RNA nucleotides called a codon. A given codon specifies a particular amino acid. The genetic code is the set of rules that relate to the base triplet sequence of DNA. The genetic code is a set of rules that relate to the base triplet sequence of DNA. The first step in protein synthesis is transcription, which occurs inside the nucleus. During transcription, the genetic information represented by the sequence of base triplets in DNA serves as a template for copying the information. During transcription, the genetic information represented by the sequence of base triplets in DNA serve as a template for copying the information. During transcription, the genetic information represented by the sequence of base triplets in DNA serves as a template for copying the information into a complementary sequence of codons in a strand of RNA. 3 kinds of RNA are made from the DNA template. Messenger RNA, which directs the synthesis of a protein. The first kind of RNA that makes the DNA template is the messenger RNA, which directs the synthesis of a protein. Ribosomal RNA, rRNA, joins with the ribosomal proteins to make ribosomes. And the third one, transfer RNA, binds to an amino acid and holds it in place on a ribosome until it is incorporated into a protein during translation. One end of the tRNA carries a specific amino acid, and the opposite end consists of a triplet of nucleotides called an anticodon. The tRNA anticodon attaches to the mRNA codon. Each of the 20 types of tRNA bind to only one of the 20 different amino acids. During transcription, bases pair in a complementary manner. The bases cytosine and guanine bind together, whereas thymine and adenine bind together. So the only difference here is uracil. And you can see that uracil is in only the complementary base sequence, not in the template base sequence. Uranol is only in the complementary RNA, the RNA, not in the DNA. Uranol is only in the RNA, and it binds with adenine. That's all I can see it binds with. These are the steps. It begins at a promoter and ends at a terminator. You can see the transcribed RNA there. B. DNA stranding is being transcribed. So the U and A, T, C, G, C, UA. Direction of transcription. The codon is this part here. RNA polymerase, RNA nucleotides, newly synthesized pre-RMA. Break indicates strand is longer than shown. Intron snipped and deleted. Functional mRNA, nuclear pore, nuclear envelope, cytoplasm. If the DNA template had the base sequence of A, G, C, T, the RNA would be U, C, A, G. If the DNA template had the base sequence A, G, C, T, the messenger RNA would be U, C, A. So it doesn't seems that every time there's an A, you put U for uracil. Immediately after transcription, the transcript includes information from both introns and exons and is called prRNA. The introns are removed by small ribonucleoproteins, and there's alternative splicing of RMA, which is a process where pre-mRNA transcribed from a gene is spliced in different ways to produce several different mRNAs. Alternative splicing is a process where the mRNA transcribed from a gene is spliced in different ways to produce different RNAs. Translation. It's a process where nucleotide sequence in an mRNA specifies the amino acid sequence of a protein. Ribosomes in the cytoplasm carry out translation. Ribosomes in the cytoplasm carry out translation. The small subunit of a ribosome has a binding site for mRNA. The large subunit has 2 binding sites for tRNA. Translation occurs in this way. The mRNA molecule binds to a ribosome. Then the mRNA nucleotide sequence specifies the amino acid sequence of a protein. Ribosomes have a binding site for mRNA and a P site and an A site for attachment of tRNA. Number 1. An mRNA molecule binds to the small ribosomal subunit at the mRNA binding site. A special tRNA called initiator tRNA binds to the start codon, which is always AUG. What is the start codon? AUG. Translation can now begin. The tRNA anticodon, UAC, attaches to the mRNA codon, AUG. AUG is also the codon for the amino acid methionine. Therefore, methionine is always the first amino acid in a growing polypeptide. Number 2. The large ribosomal subunit attaches to the small ribosomal subunit. mRNA creates a functional ribosome. The large ribosomal subunit attaches to the small ribosomal subunit. The initiator tRNA fits into the P site of the ribosome. So the small ribosomal subunit is mRNA complex, and this creates a functional ribosome. Number 3, the anticodon of another tRNA with its attached amino acid pairs with the second mRNA codon at the A site of the ribosome. So number 1, initiator tRNA attaches to the start codon. AUG is the start codon. The amino acid there is methionine, methionine. Initiator RNA, tRNA attaches to a start codon. Large and small ribosomal subunits join to form a functional ribosome. So I can see a large subunit there. And initiator tRNA fits into the P site. You can see the P site there. Number 3. Anticodon of incoming tRNA pairs with the next mRNA codon at A site. Anticodon of incoming tRNA pairs with the next messenger RNA codon at A site. So the anticodon here is AUC. You can see here, that's because the opposite of U is A, opposite of U is A again for the next one, and opposite of C is G. So that will move to there. Number 4. Amino acid on tRNA at P site forms a peptide bond with amino acid at A site. So this is the A site here, and this is the P site here. Number 5. tRNA at P site leaves ribosome. Ribosome shifts by one codon. tRNA previously at A site is now at the P site. So now we have formed a new peptide bond. The protein synthesis stops when the ribosome reaches the stop codon. And the stop codon is UAG. The start codon was I think the start codon was AUG and the stop codon is UAG. I think that's right. Let's see. Start codon is AUG. Start codon is AUG. And the stop codon, number 6, must be UAG. So this is a summary. Now we look at cell division. In somatic cell division, a nuclear division occurs called mitosis, and a cytoplasmic division called cytokinesis, to produce two daughter cells, identical daughter cells. Reproductive cell division is called meiosis. Meiosis. This is the cycle. As we start off with the telophase, anaphase, metaphase and prophase. And then we go on to G1 phase, where it duplicates its organelles. And then we go on to S phase, where the DNA and centrosome are replicated, and the G2 phase, where the cell growth continues, enzyme and other proteins are synthesized. Interphase. Consists of 3 phases: G1, S and G2. G1. The G phases are actually periods where there's no activity related to DNA duplication. The G phases are periods where there's no activity related to DNA duplication. Virtually all the cellular activities happen during G1. The S phase is the interval between G1 and G2, the last 8 hours. The G2 phase is the interval between S phase and the mitotic phase. This is where cell growth continues and proteins are synthesized. DNA replicates during the S phase. The mitotic phase. Of the cell consists of a nuclear division or mitosis and a cytoplasmic division or cytokinesis. Chromatin condenses into discrete chromosomes. In mitosis, there are 2 sets of chromosomes. One set in each separate nuclei occurs during mitosis. During prophase number 1, there's an early prophase and a late prophase. The chromatin fibers condense and shorten into chromosomes that are visible under the light microscope. Number 1. So we had, first of all, so now we're starting mitosis here, whereas this is mitosis and cytokinesis. So mitosis and cytokinesis are in somatic cell division. This is somatic cell division, where a single diploid cell divides to produce two identical diploid daughter cells. Go from interphase to prophase, early and late, So in the interphase you can see here the spindles, and you can see the centrioles. And here you can see the mitotic spindles. So we're looking at early still. In the early prophase, DNA strands move. A constricted region called a centromere holds the chromatid pair together. At the kinetochore, is a protein complex outside of each centromere. Later in the prophase, we have a mitotic spindle, which is a football-shaped assembly of microtubules that attach to the kinetochore. Later in the prophase, we have tubulins in the pentricellular material of the centrosome start to form the mitotic spindle. As the microtubules lengthen, they push the centromeres to the ends so that the spindle extends from pole to pole. The spindle is responsible for the separation of chromatids to opposite poles of the cell. During metaphase, the kinetochore microtubules align the centromeres of the chromatid pairs at the exact center of the mitotic spindle. The midpoint is the metaphase plate. Metaphase involves alignment at the midpoint. Telophase. The identical sets of chromosomes, now at opposite poles of the cell, revert to their thread-like chromatin form. The nuclear envelope forms, and the nuclei and the nucleoli reappear in the daughter nuclei. The mitotic spindle disappears. Mitotic spindle disappears.